because I'm not speaking. So we continue the video. Um, where did we stop actually?
Good quality DI
Are
water.
we here? So let me start from here.
The rehydration tray onto a shaker rocker table for 15 minutes. Note that the rehydration tray has an angled end and a straight end. Whenever you're pouring out liquids, use the straight end so that it does not slide out of the tray. Now add 2 milliliters of buffer number 2. Shake for 15 minutes and pour out. At this point, the strips are ready to run on the second dimension gel. I am going to use Biorad's Criterion precast gels, which accommodate the 11 centimeter IPG strips that we ran earlier. Prepare the gel by removing the sealing tape from the bottom of the gel and slide out the comb from the top. Rinse the wells with good quality DI water. Tap the water out into a beaker and then place the gel sideways and remove with a piece of filter paper the excess water from the wells. Be careful not to touch the bottom of the gel as it can damage the surface of the second dimension gel. We usually do this because unpolymerized acrylamide can cause anomalies in your second dimension gel if it's not removed. To remove the strip from the tray, place the forceps on the tray and gently push on the plastic backing of the gel to push the strip towards the angled edge. Lift up the strip from the right tab. I am going to rinse the strip off in running buffer that I have previously prepared. Dip 2 or 3 times. This will remove equilibration buffer number 1 and 2 that may still be on the surface of the IPG strip. Place the IPG strip gel side up. Place the plastic backing against the back of the cassette. Position the strip centered in the middle of the well with your forceps and gently push on the strip until it touches the gel. And you can tell when it's touching because you'll feel a little bit of springiness, a little bit of resistance. Don't push too hard. Now I will insert a Precision Plus standard plug, which is a broad range standard that has been embedded in agarose, into the reference well on the right side of the gel. Break off one plug, hold onto the handle and break off the spatula, and using the spatula, push out the agarose plug. For Criterion gels, I usually cut the plug into 4 slices to match the size of the well. In this case, I need 1. With forceps, take one of the slices, place on the back of the gel cassette over the reference well, and gently push the plug into the reference well. At this point, we want to seal the IPG strip and standard gel plug into the well of the gel. I will use Protein Plus Overlay Agarose. Heat in the microwave oven until it boils, and when the temperature of the agarose is no longer hot to the touch, take the hot agarose and fill up the well. And do this in one rapid action, side to side. Working quickly, use your forceps to push the IPG strip against the gel to push out any air bubbles. Agarose is going to solidify fairly fast. You can see the agarose filling in the place of the bubbles as they get pushed to the top. Small air bubbles are not an issue. Try to get some of your larger ones, and you should not have any problems for your second dimension gel. Now that the agarose is solidified, I'm ready to run the gel. In this case, I'm going to run the gel in the Criterion gel box, which holds 2 gels. In most cases, you're going to run multiple gels and you want to use a Criterion dodecacell. Remove the gel cassette and place it into the Criterion cell. Load the upper buffer chamber with running buffer. Place the lid onto the Criterion cell. Insert the leads into your power pack power supply. Program for 200 volts for 1 hour. Constant voltage. And start. The second dimension gel has now completed running. You know it is finished when the die front has reached the bottom of the gel. We're now going to remove the gel from the Criterion cassette and prepare the gel for staining. Remove the gel from the gel box, pour out the upper buffer, and open the gel by using forceps or a similar tool by pushing the forceps against the plastic cassette towards you at the top and work your way down. So fold open like so. After I open the gel cassette, I use forceps to remove the IPG strip from the surface of the gel. Starting at the bottom of the gel, gently pull the gel away from the surface of the cassette. The easiest way to do this, turn the gel upside down over the surface of the staining solution, remove a corner, and remove a corner until the gel falls into the solution. Biorad offers many different staining choices for 2D gels, ranging from the visual stains, the traditional Kamasis and silver stains, to the fluorescent stains, flamingo and cypro. In this case, I am staining this gel with BioSafe Kamasi, and the first step requires 3 10-minute rinses in water to remove SDS from the gel. We are now going to stain the gel using Biorad's BioSafe Kamasi stain. Following the 3 10-minute water washes, we're going to transfer the gel to a tray of BioSafe Kamasi, and we are going to stain the gel for 45 minutes to 1 hour. Following staining, we are going to transfer the gel from BioSafe Kamasi back into water, and we are going to destain until you get desired background. At that point, the gel will be ready for imaging. Here you can see that I have generated a gel with a clear background and well-resolved spots. I have now completed the entire process for running a 2D gel. I hope that the techniques and tips will help you when you run your next 2D gel. As you have seen, Clean Sample and the Biorad 2D Starter Kit will help you generate good quality, reproducible 2D gels. For additional information and techniques and tips on sample preparation and 2D gel electrophoresis, you can visit our Expression Proteomics website at www.expressionproteomics.com.
Okay. Did you all lesson it? Okay, so that was about basics of 2D gel. You can see how I mean it's a long process. It's a long process, but if you keep working on day night basis, it will be very habitual. So when I started doing Western blots in my PhD, I was quite yeah, it was a lot of work there, a lot of work. But I did for 3 years this Western blots on the basis of native gel, SDS page, proteomics gel, 2D gel, 3D gel. So I run all the kinds of gels a person could think of, southern blotting, northern blotting, and on daily basis. It's so I was quite used to it, and I could produce some good results for the outcome. So that was from my side, from the perspective. So there are some results of mine from these gels. So these were the results I ran for the phospho I1 antibody with concentration of H2O2 increasing 1, 10, 100, 1000 at 31 kilo Dalton base pairs with CSQ as a base, and we had this yeah, bell-shaped curve. So that was one Western blot I did. This is the Western blot done also in the presence of diamide and H2O2 with control 1, 10, 10, 100, and 1000. So it was reduced and it was non-reduced. So this was monomer and upper one was dimer. So this was 50 and 100 kilodalton. So we can see a very nice dimer formation in this. So these are the various plots that we ran with the help of 2D SDS page actually. So this is not related to you. It is my mass spectrometry data for my PhD. This one I ran some non-failing hearts, ischemic hearts, dilated cardiomyopathy with the control samples, and I could see some nice results also there with the dilated cardiomyopathy as having lots of protein presence of CTGF, whereas the non-filling have less. So we can say that the CTGF is highly present in the filling samples. So like this, I just showed you simple application now how you can use this. Uh, how could you use Western blots in various ways in various applications? And if you read some papers, you will see a lot of blots, protein working, IP, immunoprecipitation, some DNA protein interaction, protein interaction, protein isolation. So all these is only possible because of protein biology, that is your proteomics, and for that your SDS page, all these reactions are quite very much important whenever you want to do it. So keep them in your mind while doing so. So go through these reactions again one more time. So this is how the summary of the results of protein purifications. You have homogenates of all samples. So you do the salt fractionations, ion exchange chromatography, molecular exclusion chromatography, and affinity chromatography. So single band SDS page can accept it as a criterion of pure protein or polypeptide. And this way you can separate between your all samples. some summary results. Now after the mass spectrometry, something about multi-DOF. Once you've done your SDS page, you have isolated your protein in your gel. Now it's time to recognize that protein, how much size is it, which protein does it have, what is the structure of that protein, and you can do it with the help of digestion of that protein with the help of trypsin. So initially in the wet lab, you cut your DNA, your protein part with the help of trypsin as an enzyme into small peptides, and these all peptides will run through a matrix. So this is the matrix we will put our protein, so laser beam will put on it. Then afterwards they will be transferred through this ion detectors. So the ions that has been targeted of the laser beam, you can check them will have a detector, and then you can see various results, and then you can have a nice results afterwards. So here you can see some peaks showing of insulin, beta galactoglobin of 5733, 18388, various peptides of intensity. Then multi-DOF spectrometry, some results from that same part. So even in my case also, if you can just in my PhD, so I did a lot of multi-DOF actually. I did ESI, electron spray, multi-DOF in that case. So what we used to do is, so you spray of needles, your ions will move out, then at the end you will have small paths, and then you can detect them in the mass spectrometry with the high voltage charge given to them. So because of that, oh, the battery is gonna go 12%. Just a second. So redox-induced disulfide bridges and glutathione formation. So this is how you see various peptides of the protein. So this is the various amino acids group. You can see in the gray being founded out, and the white one are not founded out. So these peptides were found out, and we can see some post-translational modifications in the presence of manganese, not presence of manganese and H2O2. This is in presence of H2O2, in presence of manganese, in presence of both. So in the presence of H2O2, what I could see some persulfide and glutathione peptides present in my sample. So this is how you interpret the data. You can find some glutathione modified cysteine also in the post-translation modifications as per the results from the software peaks that we use for the ESI mass spectrometry. Where we were. Yeah, something about mass spectrometry now.
Bruker MALDI-TOF process. MALDI-TOF analysis provides additional structural information for target molecules. As MALDI-TOF is a two-phase process. Fragmentation can occur by application of higher laser energies, known as LID, laser-induced dissociation, or a background gas can be introduced to promote ion collisions. This process is known as collision-induced dissociation, or CID. Samples are co-crystallized with a suitable matrix on a sample carrier. The samples are vaporized by the laser into a vacuum while being ionized at the same time. During this process, the sample molecules can become excited. The now charged and excited particles are accelerated through the application of a high voltage, and fragmentation can occur as they travel through the flight tube. In this process, precursor ions and their respective fragments travel together at the same speed. Based on the expected time of flight of the precursor ions, Specific ion packages may be individually selected, and all other ions blocked from continuing through the flight tube. The selected ions are then re-accelerated, with the precursor and fragment ions traveling at different speeds towards the reflector detector. The ions travel via the reflector towards the detector, and thus a top-top spectrum is recorded. The detection of the selected precursor ion and its fragments creates a top-top spectrum. Whether fragmentation occurs through the use of higher laser energies or a collision gas, the structural details derived from the MALDI top-top spectrum provide a deeper analytical insight of the targeted compound.
So that was the basics of MALDI top-top. I think
Today, we rely heavily on biotechnology for many of the necessities of life. For the food we eat, the clothes we wear, fuels for transportation, earth-friendly plastics, pharmaceuticals to treat our illnesses, and dietary supplements to help us stay healthy. Many of these products are produced using three core biotechnology processes: fermentation, recovery, and purification. Fermentation is basically cell farming. We program cells to produce a product. We nurture them as they grow and reproduce, and then we harvest them. In recovery, we separate our product from the cells where they were housed. And then in purification, we go a step further by removing everything else that's contaminating our product, leaving us with a very pure, concentrated solution. In this program, we're going to look at a typical purification process used in the manufacture of GFP, green fluorescent protein. GFP is broadly used as a biological marker because it's very well tolerated by most cells and doesn't interfere with normal cellular function. We'll examine the technologies, equipment, and materials used, how to prepare for the process, and how the GFP purification process is managed, step by step. There are two main operations used within purification: chromatography and filtration. Before we can appreciate exactly what these process steps are accomplishing, we need to take a closer look at what our green fluorescent protein has been through so far. Back in recovery, our harvested cells from fermentation were homogenized. Our target protein was inside each cell, so to get at it, the cells had to be ruptured. Homogenization freed a flood of cellular components, including membrane debris, cytoplasm, DNA, and proteins, including our target protein, GFP, and they were all mixed into the buffering solution used to suspend the cells. Most of these solids were then removed through centrifugation, or by centrifuge, but the liquid that remained, the clarified lysate, was still rich in biological products, including dissolved chemicals, proteins, and other impurities. Amazingly, through purification, we can target a single protein within this biological soup. In our GFP purification process, we'll be using multiple types of column chromatography. The column is a cylinder filled with glass, ceramic, or polymeric beads, which are engineered to interact with or bind with molecules based on one or more physical properties. Chromatography relies on differences. Each molecule has a unique set of physical characteristics, such as size, charge, or extent of interaction with water. Chromatography uses these differences to separate the target protein from other proteins and chemicals. Sometimes size is used to differentiate. Some beads have small holes in them and can temporarily trap or at least slow down smaller molecules as they travel through the column of resin beads, while molecules too large to enter the pores move around the beads and exit the column first. This type of chromatography is called size exclusion. In the case of charge, opposites attract, so a negatively charged chromatography bead will attract and bind to positively charged components in the process stream. Likewise, a positively charged bead will bind negatively charged components in the process stream. This charge-based chromatography is called ion exchange chromatography. And then there's water. Molecules that readily interact with and dissolve in water are called hydrophilic, water-loving, while those that don't are called hydrophobic, water-hating. Proteins contain regions that are hydrophobic and regions that are hydrophilic. Because water tends to form a shield around the hydrophobic patches within the proteins, they are not exposed to interact with the resin beads. By adding salt to the protein solution, we remove the water shield, exposing these hydrophobic patches on the protein and resin so they can interact. This is how HIC, or hydrophobic interaction chromatography, works. In our process, which uses ion exchange and hydrophobic interaction chromatography, the chromatography equipment is housed on a skid to make it compact and mobile. The main part of the apparatus is a glass column filled with resin beads, but we also have pumps to move the clarified lysate through the process, a supply hose and port to feed the column, a pre-filter to remove any remaining particulates, usually solid cell debris that has not previously been removed, before the clarified lysate enters the column, an exit port for the processed solution, and auto switching valves for directing processed solution to either waste or collection. To help monitor the chromatography equipment and the solutions flowing through the unit during the process, a number of sensors are located along the product flow path. There is an electrical conductivity sensor at the column inlet, a pressure sensor just before the pre-filter to help identify a filter clog, a flow meter to measure the rate of solution movement through the column, and an air sensor to ensure that no air has entered the flow path. As a solution leaves the column, it passes a UV sensor that reads optical density, a second conductivity sensor, and a pH sensor that measures how acidic or basic the solution is. The conductivity sensors let us know when a new buffered solution has filled the column. When the conductivity reading on the exit of the column matches the reading from the sensor at the inlet of the column, then we know the new solution has completely displaced the old one. The ultraviolet UV sensor monitors the concentration of protein in the product by observing the optical density of the passing solution. This sensor works hand in hand with the valves on the exit of the column. Through the controller program, we can set a protein concentration threshold. When the optical density of the solution leaving the column is below the threshold, the valve directs the flow to waste. When the optical density of the solution leaving the column is at or above the threshold, this means that solution contains our purified product and the solution is directed to a collection vessel. TFF, Tangential-Flow Filtration, is at its heart a simple process step. We're going to pump a fluid through and across a special type of filter known as an ultrafiltration membrane. The size of the pores in the filter material determines what passes through and what's held back. As with almost any filtering process, we can choose what we want to keep. The solution that passes through the membrane is referred to as the permeate. Because the pores of the ultrafiltration membrane are small enough to keep the product from passing through, the permeate contains no product and is sent to waste. The portion of the feed stream that does not permeate the membrane is called the retentate. It contains the retained product and is the stream we are most interested in. What makes TFF different is a core technology that enables it to be faster, more efficient, more flexible, and even self-cleaning. In conventional or terminal filtration, a fluid is pumped directly into a filter. The particles within the stream that can't fit through the pores of the filter build up at the filter surface, eventually clogging it. In tangential flow filtration, the stream moves across the filter, that is, tangential to the filter, instead of directly at it. The cross-flow current actually picks material back out of the filter media or membrane and into the stream. This retained material, called retentate, is recirculated to the supply tank and will continue to loop through the filter for as long as the process runs. We'll be using TFF for two different tasks within the purification process: concentration and diafiltration. We are processing purified GFP from a chromatography step. The fluid from the chromatography step is purified green fluorescent protein dissolved in a buffering solution. In diafiltration, we add new buffer to the GFP retentate while displacing the old buffer, effectively exchanging buffer solutions. Note, the GFP is retained by the membrane. If we don't add a new buffer, then we're concentrating our solution. Concentration is simply the removal of water and buffer components from the feed solution that results in a more concentrated solution of green fluorescent protein. From operation to operation, or product to product, the purification process can look quite different. It could be as simple as a single chromatography step, one cycle of TFF to concentrate the product, and final filtration. Or it could involve several different types of chromatography, diafiltration between chromatography steps, and a final conventional filtration. Our green fluorescent protein process is pretty typical of the process for biopharmaceutical products. The clarified lysate will be pre-filtered, go through anion exchange chromatography, and the GFP collected and pooled. Next, ammonium sulfate will be added to make the solution high salt, followed by Hydrophobic-Interaction chromatography, HIC. The GFP is then collected and pooled again. The solution then goes through a TFF ultrafiltration step, which includes concentration of the GFP followed by diafiltration to exchange buffer solutions and remove salt. And the process finishes up with final filtration into bulk bottles or bags. It's time to gather everything we'll need for the purification process and ensure that the area and equipment are ready to go. The chromatography skid and TFF system are checked for proper operation. Process hoses are attached and examined for leaks. We verify that we are using the correct column resin and that the resin is properly packed. The column is filled with a storage buffer, and the product path is checked for trapped air and purged if necessary. Our raw materials include the clarified lysate from the recovery process, various buffer solutions that are tailored to specific process steps, and ammonium sulfate that we add to one of the buffers to make it high salt. The purification process is managed through the use of a batch record. There is a separate batch record for each processing operation. These documents lead the operator through the process step by step, with each step requiring a sign-off and separate verification by a second operator. The batch record also includes spaces for documenting times, activities, operation steps, and instrument readings. Before the process can begin, the purification area must be cleaned, disinfected, and organized. Any unnecessary equipment or materials are removed. All equipment must be cleaned, sanitized, and set up as required by standard operating procedures. All required materials and documentation must be gathered and prepared before the process may begin. The purification process begins as the transfer tank of clarified lysate from the recovery process is connected to the inlet pump on the chromatography skid. The first chromatography step in our green fluorescent protein purification process is anion exchange. At this point in the process, the pH of the clarified lysate is about 8.0, which means that the protein is negatively charged. Because it is negatively charged, GFP will bind to the positively charged anion exchange resin. The pump draws the lysate from the vessel, past the first conductivity sensor and pressure sensor, and through the 0.45 micron pre-filter. The pre-filter removes any residual cell debris or other particulates that may have contaminated the solution. If the pre-filter begins to clog, the pressure sensor at the inlet side of the filter will register a rising pressure, and the controller will signal the need for a filter change. After pre-filtering and before the column, the lysate passes through a flow meter and an air sensor. Then, as the lysate passes over the resin beads, the negatively charged protein binds to the positively charged beads. The solution leaving the column passes a UV optical density sensor, a conductivity sensor, and a pH sensor. The optical density sensor's low readings confirm that the GFP is not in the solution, so the outlet valve sends the solution to waste. When all the lysate has entered the column, or when the capacity of the beads to bind the protein has been reached, it's time for elution. Elution is the release of, in this case, green fluorescent protein from the beads by using a new solution, in this case a buffer that includes NaCl, sodium chloride solution. As the new buffer is pumped through the beads, at some point the GFP no longer binds to the beads and is released into the buffer. The resulting product stream is usually referred to as the eluate. The UV optical density sensor, which measures protein concentration, indicates when product begins eluting from the column. At this point, outlet valves are switched to allow flow of the eluate, the product stream, to a collection vessel. When the UV sensor indicates that all of the GFP has come off of the chromatography resin, the outlet valves are switched to waste. When all of the eluate has been collected and pooled, the anion exchange chromatography step is finished, and it's time for HIC, Hydrophobic-Interaction Chromatography. Hydrophobic-Interaction Chromatography is based on the principle that hydrophobic chemicals on the resin surface will bind to hydrophobic patches on the GFP protein. In order for this to occur, the resin and protein eluate have to be in a high salt environment to remove the water shielding. The salt we use is ammonium sulfate. To remove the attached GFP protein from the HIC column, we simply lower the salt concentration during elution, causing the water shielding to reform, and the GFP protein detaches from the resin into the elution stream. The protein-rich eluate is collected and pooled. The product is now ready for the last major step, TFF. At this point in the process, tangential-flow filtration will be used to concentrate and diafilter the GFP product stream. The eluate is rich in green fluorescent protein, but it's still too dilute and too high in salt. As the solution moves through the TFF apparatus, it leaves the supply tank, is pulled through a pump, past a pressure sensor, and then across the filter membrane. Everything that passes through the membrane, including the buffer solution, is known as permeate and, for this process, is sent to waste. The GFP protein is larger than the pores of the membrane and therefore is retained. The retained material, called retentate, is recirculated to the supply tank. Recirculation of the feed continues until the desired concentration of GFP is achieved. Following concentration, and while the protein solution is recirculating, a new solution, a storage buffer, is added to the feed. In effect, the protein is being washed by the flow of a new buffer solution in and the old buffer solution out. As this diafiltration step proceeds, the buffer solution that is being added to the feed replaces the buffer solution that the GFP was originally in, effectively removing any remaining salt as well. When this process is complete, the GFP solution is routed through a 0.22 micron final filter and then collected in appropriate containers, usually bottles or bags. The purification process is complete. The green fluorescent protein concentrate can now move downstream to final fill finish to be freeze-dried and packaged.
So students, you don't have to go through in details about this presentation, the last one that I gave you. It was just to show you that what is basically what are the techniques that you have learned with the chromatography, mass spectrometry, this GFP filtration, this TFF, TFT, SDS page, 2D page, SDS page, 2D preparation. So it's all in that presentation. You can see these videos. It was just to show you that how the practical use of them actually at the large scale level, how big pharma industries are working, how big pharmaceutical companies are working, how big biotechnology companies are working. So they are all working on the same principle. So yeah. So that's it for today. We are done with the protein part. Happily, finally today. It took me quite long today. But anyhow, whosoever wants to join with another class could go ahead. I have already shared these presentations with you. Just go through them. You will see all the videos in them. And please, whosoever has not subscribed to my channel, please subscribe. I have so far, 800 subscribers. So I'm just looking for 200 more subscribers. My channel will start monetizing then actually. So I'm in need of it. Like it will really helpful me. It's on 804 subscribers at the moment. So please do it. Please do this kind help to me. I will be very happy. On the other hand, if you want to start your channel also, I will be very happy to help you to boost you up. So on that note, thank you very much. If you have any questions, please feel free ask to me in the messages box, and then we can keep in touch then. Yeah. Okay then. Take care. Bye bye. Sorry for the glitch in the afternoon in that part.